So here I get to pretend to be Ed. But last night I had the best sleep of the entire trip down under, so I have no excuse for nodding off while I'm droning on up here. So, whereas you guys, I understand. Okay, so Ed's talk is, why is GBS complicated? Tools for filtering and error correction. So one of the motivations for developing GBS for maize, and also why it's complicated, is there's so much diversity in maize. Any two randomly chosen inbred lines of maize are more diverse than humans are from chimpanzees. So there's a lot of diversity there. And traditional SNP assays, they tend to have like a couple of PCR primers and then another single base primer. So that's a lot of primer design that's those sequences need to be conserved across all the samples that you're working with. Otherwise, you get failure of the assay. So we had lots of trying to make markers in maize lines and then having them not work outside of the panel you develop them in very well. So that led to this GBS approach, which is less dependent on the conservation of sequence to some level. I mean, it's still in there, but not as bad as traditional SNP markers. So these are Ed's big on expectations. So just starting out GBS, he did all his back of the envelope stuff, and he knew what he would expect to see coming out of the preliminary results. So he knew when it didn't come out that way, either something was wrong in the lab or something was wrong with the code, or there was something that he didn't take into account that he needed to figure out. So he's always doing that sort of game of back in the envelope. So we started out with 64 base tags working at that level, and so Ed made calculations what he expected in maize based on what he thinks, knowing his organism. So we felt we'd have something like 15% of the tags should be highly repetitive and not very useful, and half the genome is not shared between two lines, so we expect half of our GBS tags to be presence-absence markers. And then there'd be a low copy shared proportion, and with a 1% sequence divergent, expect that in a biparental population, about half that 64 tags should be segregating. And in an association population, most of them will have a mutation somewhere across maize, so most tags will have some SNPs in them in an association panel. So here's just a little plot showing the same thing, really. In a biparental, half the tags will be presence-absence. 17% will be biallelic, some will be too repetitive, and some will be not segregating at all. And then across the species, in a more association context, there will be a lot more that are multi-allelic tags. So what about sequencing errors? So just assuming, for sake of argument, that basic error is around 1% in Illumina sequencing. One thing with GBS is that, in Illumina, you get more errors further down the read, right, at the ends of the reads compared to the starts. And GBS puts all the reads at the same starting position. All the reads within a tag locus have the same starting position. So that means compared to whole genome sequence where the reads are kind of random, so the position of any given sequence varies from, any given base varies from read to read. GBS puts them all in the same place. So, but one thing is sometimes you get short fragments where you get overlapping reads in two directions. So you can the reverse reads can correct mistakes in the forward reads, and also the errors are going to be consistent. You can model them. So we're moving towards using machine learning to keep track of the characteristics of any SNPs that we call. So we'll have a whole list of characteristics. I'm going to show you that list later. One of them could be position in the read, and we get a good idea when to throw them out or not. So given that there's a 1% error rate and 64 base sequences, we expect only about half the reads to have no sequencing errors. So half the reads in your FASTQ file will have no sequencing errors, but the other half will. So there's sequencing errors everywhere. We still want to use that stuff. But these numbers aren't quite right because the errors are autocorrelated. Once you get one error, you tend to get another error in the same sequence. So do they matter, these errors? Are they 1% error rate? Well, in maize we do lots of sequences, or sometimes people might not do 10,000 lines, like we're up to 50,000 lines now. But at the time this slide was made, it was 10,000 lines. And but some people also do higher depth GBS, like with a 6-base cutter, and they get a lot more reads, so you start to see the errors. So even if, so assuming that you've genotyped 10,000 lines at half X coverage, and say you're at a particular base and there's actually no SNP there, the base is supposed to be an A, you'll still see the CGT about 17 times each. And those are all sequencing errors. So once you sequence enough stuff and get enough read depth, you pretty much got a sequencing error at every position. And we filter out the really rare stuff from the start. But whereas if there is a SNP and it's an AC SNP and the C is 1% frequency, then we'll see this kind of distribution of the different calls there, and only 50 of these will be real, and another 17 will be sequencing errors. So the errors are there because we have so much sequencing depth over so many individuals. Do the errors matter? They matter when you're trying to impute things or reconstruct haplotypes and maybe do a really fine scale analysis like that. If you're doing GWAS, they're probably not going to matter that much for the more common alleles where you already have a lot of power, but they're going to mess with your power on the more low frequency SNPs. And if you're doing, according to Ed, if you're doing genomic selection or just estimating genetic distance or doing mapping in biparental populations, the errors don't matter that much. For genomic selection, they can probably handle a fair bit of slop. Genetic distance, I think that they average out. There's just a little bit of noise, but there's so much signal. And in biparental populations, we can correct those things using the method, HMM method I showed you yesterday. So we can fix the errors. Any comments or questions? Okay.
I'm wondering about the fact that there was matter for haplotype reconstruction, and a question I wanted to ask in the Q&A before was whether or not the SNP calling pipeline to actually haplotype SNPs together, when you've got more than one SNP in one rig, 64 base pair, you can call an haplotype instead of a single SNP.
Yes, you could. And actually the first GBS pipeline worked at the tag haplotype level, and then we switched to SNPs. The problem was that if those are errors, then you basically can't use that tag anymore. And we wanted to be able to call the SNPs within them so that the good SNP is still useful, and we still get the depth on the good SNP across multiple tags that aren't even necessarily the same, if that makes sense.
With your logging output in the current SNP caller, you have you have locus information, so you could use that to easily stitch together those haplotypes, right?
Yeah, you could, or you could pull it right out of the TOPM, right? Everything that aligns together. But we don't necessarily have tools in TASSEL to do that. But it would be really easy to add that. Yeah.
I'm just thinking about one application for your polyploid species with allo polyploids, where you can actually break down to subgene SNPs and real SNPs would actually be a cool application.
Yeah, I think when you're starting to go with the haplotype level, you might want to get rid of the more rare tags, as long as you're not that focused on rare alleles, and the ones that are more common, you'd be more confident in them, that they're not just... Yeah, in biparental it's no problem because you have two common alleles that you expect, but we didn't necessarily want to throw away those other tags, so we moved to the SNP level.
Just wondering if multi-parental mapping populations of errors are so that are as trivial and kind of thinking maybe not.
Well, from I'm also, some of these slides are getting outdated, and I even think in imputation, the way we approach imputation is try to find IBD segments that are homozygous. So even across unrelated stuff, even heterozygous stuff, there'll be some homozygosity there, and you can find these segments, and you can see the errors then if SNPs that are they look IBD above some threshold, And if you get a collection of those for that particular segment, for that particular haplotype across individuals, you can start to see where the error-prone SNPs are and just set those to missing or set them to major allele or something like that. So that's even harder than the multi-parent situation. But so I think the multi-parent situation is pretty similar to the biparental, really, once you have enough depth there on the parents and that. Okay, so what do we expect for real SNPs? At least in maize, we expect the vast majority of the SNPs to be biallelic, so we use that. We get so many biallelic, we don't worry about triallelic stuff. And we expect that homozygosity will be predicted by the inbreeding coefficient, and we have lots of inbred lines in maize, so we use that to help us filter the SNPs. Of course, the allele frequency is constrained in a mapping population. We expect 1-to-1 segregation or 3-to-1, or depending on the population. And in these structured populations, we expect high levels of LD, so we expect that the SNPs will be in LD with their neighbors, so we can use all those things to filter SNPs. So, and biparentals give us a lot of power to find errors and also quantify how much errors there is in GBS, because we have this limited range of alleles and expected allele frequencies and high LD. So in maize rills, our biparental rills, like the NAM population I talked about yesterday, didn't I? I think so. Mm-hmm. And so it's all becoming a blur. And so the allele frequency, we expect it to either be segregating or not. And the 50% is the expected allele frequency. Of course, from sampling error and such, you don't get exactly 50. And we expect that the sites should be in very high LD in this biparental population. And since we have NAM, we have 25 populations. We can test pretty much every site that we have for errors, and I'll explain that with a diagram here. So, okay, so here is the allele frequency in NAM families for the SNPs in one of our previous builds. So in any given family, since we discovered the SNPs in about 30,000 taxa at this time, we expect, so we have a lot of SNPs with rare alleles, then in any given pair of individuals, parents, a lot of them won't be segregating, so only a small proportion will be segregating. So they're here, you can, I'm sure at the back of the room you can't see, there's a little distribution here of segregating SNPs, so most of them are not segregating at all. So if they're segregating, in the biparental family, we can't really see the errors in here. The error in here will just shift it a little bit, and it's buried in this distribution. But if they're not segregating in a biparental family, then we expect them to have 0. The minor allele frequency in that family should be 0. And when it's not, when it's a little bit off of 0, so when things are significantly deviating from our expected segregation ratio, but there's some polymorphism there, those polymorphisms are error. So we calculate error rate using our 25 biparental NAM populations by just excluding the stuff that's properly segregating, taking the stuff that's not segregating, and then summing this little error bump here over the total of the non-segregating stuff. So the vast majority is sitting right with no polymorphisms. So that means the vast majority, we're not seeing any errors. So we get really low error rates. If you calculate a median error, what is the median error rate here? So ignore all this stuff and go from here down. 0. The median error rate is 0. And this is across 5,000 maize lines. So.
What about spontaneous mutation?
You can't really, they're so rare that you can't tell those. Yeah, I mean, some of these may not be errors, but I think, yeah, so like when we're only seeing one polymorphism, it could be a new mutation, and we can't tell that apart from errors.
I think about maize so much as things like parasitic nematodes and things like that.
With a high mutation rates or something. Yeah, it's going to be really hard to tell new mutations and really rare alleles from errors.
So if you had 3 generations
because you'd see it again, right? So you'd see it twice. Yeah, that's a good idea. So you might do that in nematodes. It probably wouldn't be that hard to see it transmitted through the generations. So then if we take this graph and just exclude, the next one's just going to exclude this majority of things that are non-segregating, just to see the segregation pattern. So here's the segregating stuff. It's no errors, or errors are unknown. We can't measure them. And here's most of the stuff with error is low frequency error, and then there's this kind of bump in our section in here with high error SNPs. So they're the ones we really want to filter out. So we use the biparental populations, the NAM family, to help us diagnose those, detect those, and then filter them out.
This is all where there's no presence-absence variation.
Pardon me?
This is all where there's no presence-absence variation.
Yes, because we didn't, when we run it through the pipeline, we don't call that stuff at first. We just call SNPs. So the presence absence we throw on later. I'll be talking about that at the end of the talk here. Okay, so these are calculation of the error rates, and the median is 0. This is a log scale, so most of them have really low error rates, but there is a tail of higher error rate stuff that we use these populations to help us diagnose and filter out. Okay, so the TASSEL 3 has some tools to help you do some more filtering. So Rob walked you through to get to the top there, that green bit that says HapMap that's cut off. That's the genotypes. We get the initial genotypes out. Then Rob had like an arrow that just said filter, and these are the filters that are available in TASSEL 3. So you can get filtered genotypes after that. So the top one, merge duplicate SNPs. So that's when you have 2 tag loci on the ends of a short fragment, and they overlap in the middle, and they call the same SNP twice. Initially we'd get 2 different SNPs for that in the HapMap file, and then this plugin will merge those, and then
2 SNPs that are the same at the
same position, yeah.
So it looks like there's 2 entries.
2 entries with the same position. So the merged duplicate SNPs will merge those, and that's good that gives higher coverage for those and more data. The HapMap filters you can use site coverage and taxa coverage, or the inbreeding coefficient, which are already cutoffs in the SNP calling, but you can do more here again. And this also can use LD. If you have like a biparental population and you want to do an LD filter, that's really useful. Biparental error correction is how we use, it's a bit customized to our situation with NAM, with multiple biparentals. So if you have that available, multiple biparental real families especially, this plugin is really powerful to diagnose those error-prone SNPs and remove them. And then merge identical taxa plugin is what lets you merge things that have the same name. So if we call things B73, but they're on different, we've run them in multiple times, we can use this to merge them together. So once you get that filtered thing, then you could be ready with that without even imputing to go and estimate kinship or genetic distance, construct a phylogeny, calculate LD, or do genomic selection. But if you want to do GWAS, you're going to have to do some kind of imputation, or sometimes even imputation and phasing. So I guess the key point is to use the biology of your system to filter SNPs. So we're fortunate in maize to have inbred lines, the biparental populations of NAM to help us filter our SNPs. So Hardy-Weinberg, disequilibrium or equilibrium, depending on your situation, is really useful. Again, we have the inbreeding filter is really helpful for getting rid of paralogs. So it's not only we can put a really high filter for the inbred samples in our build. We expect FIT to be 0.8 or higher. So that's not saying we expect it to be 0. So we can really powerfully recognize paralogs that way. And okay, how to deal with the low coverage. What's Ed talking about here? Oh, okay, what he's saying here is even when you have 1x coverage, you have some subset of the sample that have 2x coverage or more, and you could focus on those ones to estimate FIT or FIS and apply the filters. So
What would happen if you used a species that, you know, you knew the inbreeding rate was 25% or 12.5%? Would that work?
yes, you just have to lower the min-F parameter. And
And the other one is, you keep on talking biparental populations, we would call them full SIB families, I think.
right.
Is any thought been given to half-SIB families, using half-SIB family information?
Ed talks about that here and makes a plug for using half-SIB families as like you'd have expected frequency. But I haven't quite figured that out yet because you've got all this stuff coming in from the pollen, right? So, and you don't at any frequency, right? So your expectation is you're going to have a set of SNPs that come from the mother and they're with a defined frequency, but stuff from the pollen or that's a tree example or a plant example, stuff from the where you don't know what the frequency is. So it depends how you do the crossing. If it's totally open pollinated, then you don't know, you don't have an expectation of what the frequency are for those SNPs, right? You're thinking, maybe you can answer it.
You know, we chuck rams out with 100 ewes and they're essentially drawn from a random population of the rams to the ewes.
Right, so if you genotype the ewes, then you have an expectation from that. So you can use that.
It's been able to genotype the possible parents, which is fine in a situation like that. It's a little bit difficult if you're doing forestry.
Yeah, I think I'm coming from a, before I was in corn, I was in a tree background, so I think in trees, so I think, well, there's just pollen come from anywhere, so who knows what the frequency should be, you know, so, but yeah, if you have a close population of parents, then you should genotype those parents and preferably at higher depth. It doesn't have to be whole genome sequencing, but you could do the GBS at higher depth and really know what your allele frequency distribution is there. That's going to help you as a filter. So I think Ed's main thing was use the biology of your system, and that's a perfect example.
Are there ways for incorporating, we have combinations of full and half SIB families, but they're controlled mating, so every family is a full SIB, but some males will be used twice and some females, because we have complete control.
Right.
So are there ways to incorporate, there's a bunch of families, but there's a pedigree among them, right? So sort of incorporate.
Well, are there ways? Absolutely there are ways, but have we coded those ways? Not necessarily, but that's where you'd want to take the code and modify it for your situation. I mean, use whatever, that's the point, I guess, here, which is good. So I'm really glad for these questions that help me clarify what I'm trying to say, which is that, yeah, use what you know to help you identify what's a good and what's a bad SNP. Because with GBS, especially in maize, we have all this porology and things, so we get SNPs that aren't very good, you know, so we need to filter them out. So GBS, there's noise in there with the signal, and the cleaner the signal you can get, the better. So use your own biology to separate those things.
So you got about 15 minutes before tea. What I'm going to suggest for you, Jeff, if you finish this one, I know you've got 2 talks to do in this set.
Yeah.
Let's do this one, have tea, and come back to the imputation.
That sounds good. Makes a natural divide.
Just
Yeah.
easy as.
Yeah.
Okay.
And you know how long these talks take depend on how many questions there are, but the more questions there are, the better it is. So,
Yep.
so if I don't get through everything, it's fine. So after filtering and using the biparental stuff to get error rates, we see that our homozygous error rate of calling one homozygote another homozygote is really low. And this symbol didn't come out right, but this is when it's really heterozygous and we call it a homozygote, that's really high error. Even though we have RILs, they're not completely homozygous. So we have sections we know are supposed to be heterozygous, and we expect it going in. We know we have low coverage, so we're flipping, when it's a heterozygote, we're flipping a coin. So we get it wrong a lot of the times. It doesn't alarm us. It's what we expect.
So this is what we typically term this as undercalling the hets, right? We undercall the hets.
Right, that's right. And then after we filtered using the minimum F and such to identify paralogs, we get rid of most of the SNPs that are in the wrong place. So So that's, we keep that to a minimum. So then we can compare. So I showed you about calculating error rates with the 25 NAM families. We could also do it with external data such as the maize 50K SNP chip that I mentioned. Some APK1 sequence went into that. So
You want me to give the synopsis real quick? Real quick. So
no, there's not enough time. Forget it. So there is some, because we use APK1 in that, developing some of those SNPs that went on that chip, there's a lot of stuff in common.
HapMap1 SNPs overlap. That's where those, then they went on the SNP chip.
Yes.
That's it.
Okay. So that gives about more than 7,000 SNPs in common, and we have the 279 of the maize 282 association panel in common, so we can look for error rate comparisons. When you use all genotypic comparisons, it's about 1% error rate. If you use homozygotes only and compare whether they were flipped or not, the error rate goes down. So it's pretty decent. It's not perfect, and we don't know which one's right. Is the GBS right or the SNP chip right? So another way we can get error is just when we repeated a sample, like we've run B73 multiple times. We've run a lot of other lines multiple times. So we recognize that and calculate an error rate, and that's really low. That's the internal GBS error rate. So that's like 3 different ways of calculating errors. So Ed thinks that like maybe half these 50K chip errors are from perology, that they think they're scoring this position on the genome, but they're actually scoring a different one. So I don't know, is it GBS? It's both, or is it the SNP chip? Is it GBS or is it the SNP chip? It's probably both. So another source of error is alignment. So when you use a couple of aligners, you can get, they don't always agree on what the best unique position is. If you use so these two, half the time about, they agree. You use 3 aligners, the agreement is down less. And if you use 4 aligners, it goes down even further. So which position is the right one? So when you have misalignment, that introduces artifactual SNPs, introduces errors into the system. So which alignment is real? So the way we're handling this is we're using genetics to figure out which one's real. So we're using genetic segregation. So Faye has been working on that. Great hair, isn't it? And so the way we do that is we can, I actually showed you yesterday, is follow the segregation of a tag as a presence-absence marker and then use that in a framework map, whether that's a 50K SNP map or GBS. Since we have GBS on 30,000 lines, we can use that. So we can do that in both the linkage context, which means in NAM basically, or an association context where we do like an association mapping of the presence-absence of the tag across all the samples. So doing this, taking each GBS tag and using it as a trait and mapping it, LD mapping it or linkage mapping it, that involves trillions of tests. So Faye did a lot of work making those tests really fast and then running them on a supercomputer in Texas. And so he has a paper in review on this work. So the idea is to put all the SNPs into the discovery TOPM and then look at all the features of these SNPs and train a machine learning model that will then tell us what SNPs are good and what SNPs are bad based on all their features and all the knowledge we can put into it. Then filter that TOPM down into a production TOPM that we distribute for everyone to apply in maize.
Yeah. Could you, in the discovery, before you use the TOPM for production, put some already known SNPs that you know from literature, previous studies, some things like that you actually get into your production pipeline, some previous knowledge that maybe you maybe the filtering of, if there's a low correlation, you would consider it as an error, but because you've seen it some other times, you could actually say it.
So the question is, could you add information into the TOPM on previously known SNPs and then use that in a production context?
Yes, that's a great idea. We should do that.
So I would say there's probably one caveat, right? Like, so there's all kinds of SNPs in the literature that may not have anything to do with the GBS tag.
Yeah.
And if that's the case, putting that information in the TOPM is absolutely pointless.
I guess we'd have to know their position in their current genome coordinates, which is not impossible. So,
Yeah.
Yeah, I think that's a great idea. What we're going towards is a HDF5 that holds everything and holds all kinds of annotations about each site, and we could definitely, that could be one of the annotations. This is SNP with this name or whatever in this other panel, you know. So whether it's a 50K or some other SNPs available in maize, that could definitely be done, and that would, you could pull that out as a subset. Let's compare against what we saw with the samples in common. Terrific idea. So, what do you want to filter on? That's something to add down here. So this is the list of characteristics that we're thinking about, and it's probably when we really sit down and work together on this, even within our group, we'll come up with a longer list. And so the idea is to take this discovery SNP caller and have it, when we discover SNPs, to put out all the SNPs and not really filter them much at all, and then put out all these characteristics of the SNPs in the output, and then use that as input to a machine learning algorithm, and use the NAM that gives us a subset of things we know are good or not good, and train that machine learning algorithm to tell us how to pick out the good SNPs. So that's where we're going. So absolutely, these kind of suggestions are great. If you have more ideas, send them to the TASSEL Google group. What do you want to filter SNPs on?
Yeah, we can't really plug that enough to start using that if you want to keep up with what's going on or learn about how to use this software. It's a really good resource.
So what we're trying to do with TASSEL 5 here is give the user the power to decide how they want to filter the SNPs. So we're basically going to output everything that's a SNP.
Oh, that brings up a good point. A lot of people get really giddy about employing a lot of those filters actually during the SNP calling procedures. But if you're doing a small pilot study and you want to play around with different types of filtering, just keep your filters really low during the actual SNP calling procedure, because as you saw, you can try different filters after calling your SNPs.
And some of them are the same filters that you can apply during SNP calling,
Yes.
right? So
Yes.
it gives you more flexibility.
Okay. So how much time do we have?
10 minutes.
10 minutes? Okay. Okay. So here I'm going to talk a bit about presence-absence tags. So in maize, this is the majority of the data. If you're working at the whole tag level and you're working with the more common tags, they have fewer sequencing errors in them. But we need our imputation methods to differentiate between true missing, which is biologically missing, the piece of DNA is not actually there in that individual, versus statistical missing, just a low coverage, we didn't see it. It's there, but we didn't see it because we didn't sample it. So I think I've shown this before. So there's lots of structural variation in maize compared to humans. But you can map the tag. So I talked about this yesterday, so I can kind of zip over this. So you can map the tags as long as they're in a biparental cross, say, as long as they're polymorphic between the parents. You can take this tag and totally ignore this tag, or you can map this tag when that other tag isn't there. So, and this is, I walked through this yesterday, how this is done with a binomial test for linkage. And that applies in a biparental context. It also applies in association mapping context. So this is basically what Faye did to map a large number of tags across a large number of samples. And what we like with the association mapping context is that it can give you a more precise location on the genome than linkage mapping does. So Faye developed this model, this method to map tags. So he took the list of GBS tags of interest, that's this master tags list, and he either did GWAS on all the samples we had, or he did joint linkage mapping in NAM. And if they map, then he goes further. Yes, they map genetically. Then he asks, do they align? So do they physically aligned by their sequence. If they don't align, then they're not aligned to B73, so they're insertions relative to B73. So there's something that genetically maps somewhere, but they don't align to the reference genome, so they're insertions. Or if you're looking at B73 and non-B73, and you're looking for deletions relative to B73, then it's because any given tag locus has low depth, it's not very reliable. But if you do bins, you can see regions that are larger deletions, and you can also see by higher read depth in a bin that there's been a duplication there. You can look at copy number variation. So Faye's done a lot of work in this area. So I use machine learning to help them. So the problem when you're doing GWAS is that it's affected by structure, right? By population genetic structure. It can give you false positives. But to account for population genetic structure, it's generally done using a mixed linear model, which takes a lot of time to compute. So, but Faye needed to do trillions of tests, as I told you. So he needed to account for structure in a different way. So he used machine learning to help him account for structure. So he took these 26 million tags, and he did GWAS and joint linkage and NAM, and he ended up with ones that were mapped in either or both. And then he put them through model training, and I don't remember what I think G is the ones that map with GWAS, GJ is the ones that map with both, and J was joint linkage only. He put this through model training using like the B73 tags as like a positive control, and came up with a machine learning model that let him filter out of these 26 million. He ended up with 4.5 million tags that he felt were mapped reliably. So then he studied those. So using the B73 tags as a positive control, he could show that 95% of the time he could map them within 1 megabase of their true position, and 50% of the time within 10 kb. So he has these 4.5 million tags mapped in maize to pretty good precision. So this mapping of tags is like a framework of the maize pan-genome. So we want to go past having just B73 in order to really capture all the structural variation. We're trying to reconstruct genomes on multiple individuals, about 12 in total, counting B73, where we're doing whole genome sequencing. Then we're going to make de novo contigs and try to construct those genomes. And if we have this genetic segregation information on 4.5 million GBS tags, we have an idea of where they go genetically, so we can use that to help guide the physical assembly of these pan-genomes. So we encourage the Java programmers or people that want to be Java programmers that want to take the pipeline and add things to it, and especially if they want to share those with the community, to become a power user. Play with the code to add your features. We've already had several examples of what people would like. If you're capable of doing this, we highly encourage it. We'd like to crowdsource this as much as possible. So there's documentation about the code. The code is at SourceForge. It's all open source. There's Ed's been working on a guide to programming TASSEL. So when you're like, where do I start? You can start here, so it's not, you have to figure it all out from scratch. There's a lot of explanation of how we code in TASSEL, how it's structured, and that there. And of course, you can contact us when you have questions. And when you have questions, there's a Google group. The Google group is not just for the power users, it's for all users of TASSEL, and not even TASSEL GBS. But there you can post, first of all, go to the Google group and look for the answer to the question you have.
Search your question.
If you don't find it with your first search, try an alternative search, and then if you still don't find it, go ahead and ask the question. And so we kind of partition these questions according to our different expertise.
And I would just comment that in the recent activity, I've been noticing a lot of users participating in answering questions as well.
Yes, I'm
So
very
it's
grateful for that.
Yeah.
And that's all I have on this part.
There's a question in the back.
Oh.
Yeah.
So those 4.5 million tags of phase that went through that machine learning process, did he go back and check what proportion of those were mapped So in other words, what's the correlation between that circular maps across all the liners and the ones that were machine learned and found to be the best location?
That's a very good point, and he did that. I don't have the numbers, but it really launches me into something I should have said, that where we're going with TASSEL 5 and the TOPM is not just to store one position from one aligner, but to store multiple positions from the multiple aligners, and then store what the genetics say, so we can choose the position that the genetics support as well. So that's kind of where all this is going, that we can use all information together and use the genetics to help us sort out which is best, and then pick that best one.
I don't remember the numbers either, but I think the take-home message from that analysis, and correct me if I'm wrong, Jeff, because I just saw it at one lab meeting, but it was that if you store all the top alignments from the aligners, typically the genetically correct alignment position is in the list of the top alignments, but it's not necessarily the first one, and not even very often the first one.
So when you say the top aligners, were you scoring the top 3 or top 5 or something?
This is the detail that I don't remember. I know that they were doing top 10 for us.
Sorry, not Ed, but Faye kept from the different liner the top 5 positions, and almost always, she's absolutely right, almost always the right position that from genetics was in that list, but it wasn't always number 1. So.
Which is why they're now treating each alignment as simply a hypothesis.
Right, the
To be
physical
tested.
alignment of a tag is a hypothesis, but if we lift that 64 base limit, it's going to be a much better hypothesis, so that's a really good reason.